welcome everyone to the January PathSafe webinar. So my name is Elaine and I'm a Senior Project Manager on the PathSafe programme, and I'll be chairing today's session. So for this webinar today, I'm delighted to say that we're joined by colleagues from Workstream 2D within the programme, specifically Dr. Fran Collis from the University of Oxford, who is leading on this project. So Fran is a microbiologist who began her career working in veterinary and clinical labs before moving to research. She then moved on to complete a doctorate in Martin Maiden's lab at University of Oxford and has been researching Campylobacter ever since. So Fran is here to talk today about their team's work on Campylobacter as part of PathSafe. Could I just get people to go on mute, please, everyone, just to ensure they're muted? Thank you. Sorry about that, Fran. Yes, Fran is here to talk today about the team's work on Campylobacter as part of PathSafe. So this project is entitled The Genomics of Antimicrobial Resistant Campylobacter Transmission Through the UK Agri-Food Systems, and the project is focused on investigating just that. So work started in February last year. And at this webinar today, Fran will update on progress to date, ongoing work and ambitions for the future of the project. So just very briefly before I hand over to Fran, I'll cover off a couple of housekeeping items. So just to note that we are recording the session today and that we plan to make a copy of the recording available on the PathSafe website in due course. The session will last no more than 1 hour and we'll ensure that there's time left at the end for questions. We ask that you keep yourself on mute while the speakers are presenting, and please feel free to add your questions in the Q&A bar during the talk as we move through, and we'll come to questions at the end. So we'll come to you, ask you to unmute so you can ask your question to the team, or you can raise your hand during the Q&A at the end and we'll come to you. And any questions that we don't get to answer, we will take away and respond afterwards. So without further ado, Fran, I will hand over to you to kick off.
Thank you very much, Elaine. Thank you for the introduction and the invite to speak today. Hello, everyone. I'm just, hopefully you can see this. Can you, the slideshow?
Yes, yeah.
Great, brilliant. So as Elaine said, this is the subject I'm talking about today. I've pitched it towards a general audience, so hopefully there's something for everybody. And like Elaine said, if you do have any questions afterwards, please don't hesitate to contact me, and this is my email address here. So for this project, as part of the PathSafe programme, we set out to sequence up to 2,500 disease isolates from Oxfordshire, and some from Wales as well, and then up to 1,000 agri-food isolates, and particularly concentrating on those that are less representative in culture collections that currently exist. And then for their outcomes, we want to generate a representative, culture collection, and use this then to sort of do more in-depth analyses such as source attribution, genome-wide association studies, and so on. We would like to determine the predictive mechanisms of AMR from the genome, and look at longitudinal trends and risks to human health. And then I'll be talking about a data set from human disease that spans the COVID lockdowns in 2020. So this is, if you like, a natural experiment or opportunity to see how the effects of reduced eating out, foreign travel and so on impact on the cases of human disease. And then finally, working in Martin Maiden's group, we run the PubMLST database in Oxford, and so we're working as another part of the programme to integrate PubMLST with the PathSafe resources. So for the purposes of the talk today, I'll briefly talk about why we're interested in Campylobacter and talk about a previous study funded by the FSA that we did looking at source attribution, but for isolates from 2015 to 18. And then I'll give you an overview of the current study results, and I will emphasise that these are preliminary because we're still receiving sequencing data back at the moment. But I will talk specifically about the Oxfordshire human disease isolates that we have, and talk a bit about the source attribution and antimicrobial resistance that we're seeing, and then finally ongoing work that we're doing. So we're really happy that we have the opportunity here to work with Campylobacter as part of this programme. It causes severe gastroenteritis. It's the most common bacterial cause worldwide. It causes in the UK approximately 600,000 cases a year, but you can, it's thought that there's 10 times this number out in the community, in fact. And it's more common than most of the other foodborne diseases put together. It can have some horrible side effects. Guillain-Barré syndrome is a temporary paralysis. It can be associated with reactive arthritis, irritable bowel syndrome, and there's a huge cost to the economy every year. And also it's on the WHO watch list for antimicrobial resistance, particularly for its increasing resistance to fluoroquinolones. So it's a tricky organism to work with, and I guess one of the things is it's a zoonotic organism, and we can find it pretty much anywhere we look. If we're looking in the guts of animals, and also that fecal contamination of environmental sources. So, for example, environmental waters, soils, we can isolate Campylobacter readily from those. From all of the livestock, wildlife, and companion animals. And the reason we particularly focus on human disease isolates in Oxford, and we have this long-going study where we take every isolate that comes through the John Radcliffe Hospital in Oxford, and then we sequence type them. We're interested in doing this because unfortunately humans seem to be a good proxy for sampling whatever's out in the environment. We seem to get whatever's going. And of course, in this talk, I am inferring to the large part from human disease using the source attribution studies where these Campylobacter isolates and sources of infection are coming from. But this is substantiated by large isolate collections from livestock, animal sources, and so on. So, as I mentioned, there's a previous source attribution study that was funded by the Food Standards Agency. It's published, so you can reach it by this link at the bottom here. For this study, we were looking at human disease isolates again, but supplementing the Oxfordshire disease collection, we also had another site in North East England, and this was covering the years 2015 to 18. And you see there's more than 5,000 isolates there. And then, as I said, we were working with the APHA, sort of large livestock and retail meat survey collections from duck, sheep, cattle, pig, turkey, chicken. So again, nearly 4,000 isolates. And I'm just going to show you a couple of highlights from this report as a starting point for where our current project is kicking off from. So here we're looking at source attribution. So modelling where human disease is coming from. And in the figure on top left there, chicken shown in red is accounting for approximately 80% or 60 to 80% of human disease. And this is since 2004 up until 2018 in this study. There's also significant numbers from ruminants, and it's difficult at this point in time to distinguish between cattle and sheep. And then yellow, you can see a very small line at the top from wild bird and environmental sources. So it's not to say that people don't get infections from those, but the signal was kind of obliterated by the sequence types that we see from chicken. And then we're also looking at antimicrobial resistance in this previous study. And we did have isolates going back to the 1990s that we included in this. So yes, there's been quite a dramatic increase in resistance over time. So we're looking at C. jejuni here. It's about 5% resistance for fluoroquinolones back in the 90s, and that's increased up to over 40%. And similarly, the resistance has increased for tetracycline over this period. Page has stopped moving. Right. And then this was the resistance levels we were seeing predicted from the genotype, supplemented in part by phenotypic MIC testing. But if you note particularly the green and the lighter blue, These are isolates from chicken abattoir and retail meat surveys. So these are the ones that have the highest fluoroquinolone resistance and also the tetracycline resistance. Also note there's high tetracycline resistance levels in pigs and ducks, but in pigs in particular, the numbers are quite small. Now we're looking at C. coli on this slide. So similarly, oh, sorry, I think C. coli in humans, this is the resistance pattern. So again, similar to C. jejuni, we're seeing an increase in resistance from 5% approximately in the 1990s up to approximately 40%. So similar levels for both organisms. We consider both because C. jejuni is causing about 90% of human disease year on year, and C. coli is about 10%, and then there's occasional other species. So for the purposes of today, I'm concentrating on these two species. With C. coli, we also see some resistance to aminoglycosides coming through there and macrolides, so a bit more resistance to multiple antibiotic classes is a feature of C. coli. And then if we look at them in animals, again, we're looking at the pale green and the pale lighter blue for chicken isolates. We see high resistance amongst C. coli isolates for fluoroquinolones and tetracycline. Again, tetracycline resistance in C. coli is quite high from pigs. There's more numbers this time because we tend to see a bit more C. coli in pigs. Quite high numbers in ducks, and then again aminoglycosides, we see some quite strong resistance in pigs. So that's where we were at in terms of sequencing the human disease isolates, but we're interested in this longitudinal ongoing study to really look at the impact of what looked like some quite promising interventions and sort of Campylobacter focused initiatives. So here, for example, taken from this report published by the UK HSA, looking at chicken meat surveys. Again, like I said, there's been initiatives to sort of reduce the contamination by Campylobacter on the most heavily contaminated chicken flocks, or chickens, sorry. So back in 2014-15, we were seeing sort of 73% of neck skin samples Campylobacter positive, and then particularly we're targeting those with highest contamination at more than 1,000 colony forming units per gram. So that was 19.4%. It's taken a lot of work. Industry, Food Standards Agency and so on have worked together, but over time, you can see this is a success story where we now see sort of 56% of neck skin samples are now Campylobacter positive. So that's a significant decrease, and 7% of neck skin samples were representing the more heavily contaminated birds. And in particular, there's higher rates or greater variation in the Campylobacter results that they're seeing amongst the smaller retail shops. So we're interested to see if that's impacted on human disease at all. And similarly, there's been real efforts to reduce antibiotic use on farms. So here's a couple of infographics that I've grabbed from the Veterinary Medicines Directorate. So huge efforts to reduce particularly critically important antibiotics. I mean, colistin here is down by 99.9%, which is amazing. And also the British Poultry Council is similarly saying they've reduced use on farms by 97% for critically important antibiotics. Sorry. So I'm now going to come on to the, as I said, preliminary results from the current study. And just to say that you can access the information for yourselves if you want to. We hold all of the isolate in the isolate collection on PubMLST. PubMLST, you can imagine, we use it. There's a box on the left there that says typing, and that's like a dictionary of all the different alleles and so on that we find. And then we also have in the isolate collection examples of where all of these sequence types have been found, and then we also have the genomes to back this up. So it's a somewhere to deposit both genomic information and also isolate metadata and combine the two. And there's lots of downstream tools you can use for analysis also. So if you want to access the work that we're doing for PathSafe, you need to go on the isolate collection, click that box there, and on the right hand side you'll see there's a drop down menu for projects, and then this is showing you an overview of the isolates that we have. So at the moment I've got information in there for just over 3,000 isolates, and we have sequencing results back for 2/3 of those. So like I say, we're still waiting for the final results to come back. But this is showing you an overview of what we have. So we have some historical samples in that we're doing for this part of this project program, and then most from human disease at the moment. So here we have an overview of the number of cases reported in Oxford. So like I said, these are the isolates coming through the John Radcliffe Hospital in Oxford. It largely mirrors what's going on the national scale, and we have tested this formally in previous studies across 3 central surveillance sites. And we know that Oxfordshire is representing approximately 1/100th of what's going on nationally when we're covering all of those sequence types that are out there on that scale. For this talk, I'm concentrating particularly on years 2019 to 2022. We will have data coming through for 2023, but they're not complete yet. And this is again an overview of the isolates we have. This isolate collection actually goes back to 2003, but I'm just showing you the later years here. So we can see in terms of numbers, there's a seasonal peak with Campylobacter. So we have more cases of human disease in the summer, and often there's a secondary peak in early autumn as well. There is a gap here. We have all the isolates in the freezer, but we had to set this project up and get it up running very quickly. And this gap here represents staff in changeover in staff and being able to access all the records and so on. And here you notice another gap, and the cases of human disease were going up very, very quickly. And so the UK HSA actually took these isolates here as part of a national survey to see what was going on. I'm going to quickly describe the nomenclature that we use to describe the genetic diversity of Campylobacter. It is very, very diverse, as you might imagine, because it's occupying so many different niches. So here we have a snapshot of a grape tree, just showing, if you like, I kind of think of it like a galaxy or also a fitness landscape. So we have lots of sequence types of Campylobacter that are very closely related, which we refer to as clonal complexes. And then we also have some that are more remote, they're sort of unique. So if we look on here, we can take any sequence that we've generated and search on PubMLST, and we can either find the existing allele or assign it a new one. And then if we look at 7-locus sequence, multi-locus sequence typing to start with, this was a historical typing system based upon what we could generate at the time. Each number there is generated from a unique sequence. So, for example, ASPE4 would always be the same sequence, but you build up a profile going across. So the 7-locus profile, and this is kind of equivalent to a shop barcode. You can just blip it and tell it apart from a different one. So in this example, we have ST45 at the top. In the next one, the sequence type, which is this 7-locus allele profile, it just differs from ST45 by 1 allele here, 42. But you can see they're very, very closely related, and they're both within clonal complex ST45. So this is a group family of closely related sequence types. And as you'll see, I'll be talking a lot about clonal complexes. And although this is kind of a slightly older method, maybe, it proves very, very important in looking at biological relevance. And if we're talking to other people about Campylobacter, if you've worked with it long enough, we know what an ST45 is and where we'd find it. And then the next one down, ST1034 there, that is an example of a wild bird derived sequence type. And as you can see, it's very different in the numbers, which in turn reflect the sequences that generate those numbers for that allelic profile. Of course, in the era of whole genome sequencing, we can do exactly the same thing. But if we're looking across the core genome MLST scheme, we would do this across 1,142 loci currently. We're also able with PubMLST to look at different alleles that are representing the different antimicrobial resistance determinants. So we can look from a sort of a large overview down to individual polymorphisms within the sequence. So I'm beginning here looking at the Oxfordshire disease isolates again, looking at the breakdown of clonal complexes. So as you'll see, not all clonal complexes are the same. However, you can see that I've got 9 clonal complexes shown here, and also some isolates that cannot be assigned to a clonal complex. So they're ones that are singletons and on their own. But they're accounting between them, these 9 clonal complexes, for between 70 to 80% of human disease. And this is constant going back 10 or so years, and it will be beyond as well. I'm just showing you this for clarity. ST21 complex is probably the most predominant. And although we're seeing sort of stability long term, there are some nuances. For example, in 2015, it looks as though ST48 clonal complex is slightly more prevalent. We're seeing the one in bright pink at the top there, ST43 clonal complex, has sort of made more of an appearance in later years. ST257 shown in yellow here, that's an interesting one that's potentially sort of declining in importance. And then finally in 2021, if you remember, that was when there was a big peak. We're seeing in Oxfordshire an increase in that 464 clonal complex. So here is the same information shown in a slightly different format, but what I'm doing again is here you can see generally that the clonal complexes are very consistent year on year through this study, with some small nuances like 464 is higher in 2021. What I've done for the purposes of this talk is beginning to look at source attribution. We know from long years of research in Campylobacter which clonal complexes are most commonly associated with which source, but we will go on to test this formally. But if I just overwrite these on this figure now, you'll see that these are the most common clonal complexes that we're seeing in human disease, and all of the most common ones are coming from chicken sources. And we do, with the exceptions of ST21, which is a clonal complex, which is we describe as a generalist. So this is one that we see very commonly against across multiple livestock sources. Chances are still very, very dominant in chicken sources. It's just with our current attribution models, it's difficult to tell exactly what's a chicken ST21 and what's a cattle one. And ST45 similarly is found very commonly amongst different host sources, but this one I think particularly is very common, relatively common compared to others in environmental water sources and so on. And then it's not until down here that we have the ruminant associated clonal complexes, and this one is also one of the more common C. jejuni clonal complexes that we might pick up against pigs. So here is the sort of methodology that we use for the more formal sort of machine learning approaches to source attribution modelling, and I will be doing this when I've got the final data set. But the general gist is that if we say, for example, have a red type of Campylobacter that we've isolated from human disease, based upon existing livestock sampling data sets that are chosen to be representative, we then use leave one out type approaches to model and predict which is the most likely source of that red Campylobacter has come from. And then I was interested to look at seasonality of the different clonal complexes. And if you remember, I was in particular interested in the lockdowns where COVID-19 associated lockdowns and whether there's been any impact on prevalence of different clonal complexes, and perhaps we might see some more wild bird associated complexes if people are out walking a bit more. Yeah, that was an idea anyway, hypothesis. So the only one I've seen of note, and we also saw this in the previous study, is ST45 complex, which has a seasonal peak in the summer, and we see this year on year. When we look at the other clonal complexes, there seems to be a bit of a mixture of what's going on and quite random. And I can't see anything that jumps out to say anything particularly unusual is going on in 2020. So when I was looking particularly for the environmental or wild bird associated clonal complexes that may reflect people being out and about in the countryside a bit more, I remember it being very nice year, warm, and meeting lots of people out walking, obviously when restrictions allow. But yes, there's literally 5 sequence isolates that I can see that might reflect this, so I can't even show it on a graph. So I think this fits with Campylobacter being a foodborne disease, and reflects our exposure to the meats that we're eating, and this is at the moment the predominant source that we're seeing. And then as an aside, a lot of my research I've done looking at chicken flocks, and so the sort of stochastic nature of the previous slide, the clonal complexes that we're picking up in human disease, reminds me a lot of what I'm seeing in chicken flocks. So here, each of the different colours reflects a different Campylobacter lineage, done by a slightly different sequencing technique, typing technique on this slide, but the principle is the same. And each bar there is showing the Campylobacter variants that we might detect in the chicken flock in that week. And this particular chicken flock is a breeder, broiler breeder, meat chicken breeding flock. The reason being is that we can follow the turnover of the variants of Campylobacter for a longer period of time. And yeah, we can see that some variants are there for quite a long time and others are not. And if you can imagine that these might be filtering through to human disease, there's a bit of an element of chance in there, I would suggest. But I'm going to move on for the last piece of the talk to talk about the genomics of the antimicrobial resistant Campylobacter we're seeing in the Oxfordshire disease collection. So starting with Jejuni. Fluoroquinolone resistance is shown in blue, a tetracycline in red, and I also looked for the presence of both of these antibiotics. So I've done these 2 antibiotic classes for today, and we do intend to extend this to other antibiotics going on. But for these 2 particular antibiotics, I would suggest there's a general upwards trend, sadly, particularly for those isolates that are resistant to both antibiotics. And then there's a bit more variation perhaps with the Campylobacter coli isolates, but if you note the numbers of isolates that we see are lower here. So this is, yeah, we're still seeing an increase, particularly for the both classes of resistance in the same isolates. And then when I break that down to clonal complexes, this time for C. jejuni, I'm particularly concerned to see that there's really high levels of resistance, particularly for 354 clonal complex and 464 clonal complexes. So these are, as I said, predicted from genotype, and we will be taking these out of the freezer and testing MICs. But the 353 complex is not far behind. And then in contrast, we also see ST45 complex there has very low resistance. So again, I've overlaid the sources that these clonal complexes are associated with, and the ones with the highest resistance are associated with chicken sources. But do note that there's also ST257 clonal complex that is also chicken associated and has lower antibiotic resistance. So I suppose it's not entirely surprising if we're thinking that most of human disease is coming from contaminated chicken meat, that the antibiotic resistance may also be coming from these directions. However, I think it's really a very complex situation. We've seen that people are making outstanding efforts in reducing antibiotic use on farms, and some of these lineages are more resistant than others, and so then they're not acting uniformly. And what I would suggest is that we really don't understand enough about what's going on in these farms and what's causing these lineages to be so successful in this environment. I think also this is a real opportunity for us to understand a bit more, and then hopefully as a sort of scientific community really sort of try and address these really complex issues, of which Campylobacter is particularly challenging and difficult, I would suggest, but is also very, very useful as a model organism in this situation. So here I was looking in greater detail at the fluoroquinolone resistance and how this is working. So I'm showing here amino acid sequences for DNA gyrase, which is an enzyme involved in transcription and replication of DNA. And most 99% of the resistant isolates that we saw in this human disease collection, resistant to fluoroquinolones, have this single point mutation causing a change in the amino acid sequence from threonine to isoleucine. And when I look at the individual alleles, we have 105 different alleles across, I think, about 1,700 isolates. So there's quite a bit of variability, even though they're causing this change here. The variability is seen amongst the rest of the gene. But this is the mutation that's causing the resistance to fluoroquinolone. So like I say, that T86A change was responsible for 99% of the resistance I saw, but I also saw some interesting rarities. So this was just one example we picked up in this project. Is a change, instead of T to I, it's T to A this time. Saw this in one isolate. It was an ST230, which is part of the 45 clonal complex. And this, sorry, is allele 661. So when I looked at the global collection on the PubMLST database, I was really shocked to see that this combination has also been seen in Finland, Scotland, England, as far back as 2012, up until 2018. It is still rare on the database. It's there were 71 examples of T to 86 to A change, but that's less than 0.01% of isolates on the database. So interesting. And then we have another example here, T86B. We had 2 isolates in the collection with allele 1510. And looking at PubMLST global collection, similar combination of resistance allele and sequence type has been seen also in America, Italy, in between years 2018 and 23. And then we have one isolate with a unique allele, 2298. This hasn't been seen anywhere else at all. But that T86V pattern is interesting, I think. So there's 257 examples of that on the PubMLST global collection, but 93% of these are seen in Campylobacter larry. And I have actually got some Campylobacter larry isolates that have snuck into this project from backyard chickens, and lo and behold, they have the exact same pattern and a combination of rare or common alleles. So there's some interesting things going on. And then finally, this is an example at the bottom where there's 2 point mutations, two amino acid changes this time. And this one is really commonly seen with an ST441. I can pretty much guarantee if I see that change, it will be associated with ST441. And the allele number is 45. Similarly, this has been seen in France, UK between the years 2006 and 23. And then there's just one slightly different variation with a different allele number and sequence type, again seen in European countries over previous years. So to me, this is suggesting that these lineages are actually very, very stable. There's stable associations between the antibiotic resistance patterns and clonal complexes. So then they're not sort of changing very quickly in time on farms, as far as I can see at this point in time. And this sort of contrasts to the tetracycline resistance, where I was looking at 750 isolates and they have 70 alleles, but in contrast to the fluoroquinolone I've just shown you, tetracycline alleles are not stably associated with different clonal complexes. They seem to swap about a bit more, and this probably reflects that they're often on plasmids and can be easily transferred in this way. So this is just a really quick snapshot overview, preliminary sort of analysis of the data set that we will be looking at in more detail. Alongside this, I'm working with Keith Jolly in Oxford, who develops the PubMLST database. And we've been working to try and improve antimicrobial resistance, calling it bespoke for Campylobacter. So at the moment, you can do things like click on the ID number for this isolate, and it will show you the secondary metadata. So these would be the types of codes that you would find in ResFinder and so on. And I've also run these isolates through ResFinder as a comparison. We can now search on the database for the individual variants, and you can look across the whole global data set, which is really, really amazing and very, very helpful. I intend to develop this further for the other antibiotic classes, like I said. This does take quite a lot of work to set up in the first place, but then once it's done, it's amazing. And what we would like to get to the point is where you can actually do a printout. You put your sequence in, you do a printout of an antibiotic resistance profile predicted from the genotype. But we also will plan to do a lot more work with phenotypic testing across those different allele types, and just see what's going on there. So to summarise what I've said, 80% of human disease is coming from 9-10 clonal complexes, and they seem relatively stable over time. From what I've seen so far, I haven't seen any effects in clonal complex distribution related to COVID-19 lockdowns, changes in human behaviour and so on. And the highest levels of antimicrobial resistance are found in the chicken-associated lineages that are also stable over time and globally distributed. And so what we will be doing going forwards is finalising this disease data set. We have more isolates from 2023 that we're waiting for, and some of the Agri-Food isolates as well. I'll be looking at the source attribution analysis using the machine learning approaches as we described and used in previous studies. What I would like to do with those chicken-associated lineages is use genome-wide association studies and try and see if there's any genes or reasons why these lineages might be particularly successful in surviving through processing, really, and reaching human disease, causing human disease. If there's anything in particular about those that makes it easier for them, because presumably, particularly with tetracycline resistance, there may be a fitness cost, but this we're not seeing that. And then, as I said, we'll be looking at developing PubMLST further and incorporating this with path-safe resources to predict antimicrobial resistance from genotypic data. And finally, I'd like to thank my colleagues at Oxford. We've been working with Quadram Institute, who've been doing our sequencing for us, which has been amazing. I mean, with the worst timing in the world, the Oxford sequencing service closed just as this project was starting. So we've been really incredibly thankful for David and Carl for helping us. Of course, all the samples that the clinical microbiology staff grow for us. Because they've moved over to a PCR-based system, they have to specifically go and get those samples out and grow the Campylobacter for us. I've worked very closely with poultry industry over the years, so grateful for those. Their help in providing samples, and similarly pet owners. And of course, thank you for the PathSave programme for providing the funding for this, and to Elaine as well. Thank you very much for listening. I'm happy to take any questions. I'm going to try and stop sharing this. I can work out how to do that.
Thanks, Fran. So thank you very much for that. That was really, really interesting. So we'll move on to Q&A. Anyone has any questions on the work that Fran has presented, please do shout, put up your hand. Looking at the Q&A bar, I can't see anything in there just now, but it might be we'll give people a minute to formulate their thoughts. And Ed, I can see you put up your hand.
Thanks for that talk, Fran. That was extremely interesting. One kind of small question, I suppose. I think it was in your earlier slides that you said that ST443 had kind of appeared.
Oh yeah.
In around 2019 and wasn't seen previously. Have we any idea where that's come from or why that has appeared?
That's another chicken one. I don't know why it's appeared. Often they sort of, you get some that are very dominant and some that sort of pop about in the background. It's possibly one that's been around for a while but has become more more common. Okay. But not common relative to the others. But yes, it looks to be another chicken one. So it's really interesting, isn't it, how that happens? But again, like I said, in chicken flocks, I see a turnover of types over time, and there doesn't seem to be anything in particular driving that. I've looked at sort of associations or correlations, with weather, hours of sunshine, rain, all the sort of environmental conditions. And yes, you get that seasonal peak in infections, both in people and in chickens. But other than the ST45 complex, which is more common in the summer, and perhaps that's because it's, I think of it as more of an environmentally associated one that's good at getting between different environmental hosts, and maybe it finds that easier in the summer, maybe. That's the only one that I really can predict to any extent what's going to happen. So yeah, it's interesting, isn't it?
Yeah, really. Do you ever see any difference between kind of free range chickens and barn raised chickens? If they're more exposed to the environment?
Question. Yeah, that's a really good question. And this is why we're looking at some of those other sort of isolate collections that I've got in the freezers in Oxford, and some of those are free range. I think you may get some differences. Certainly I see sort of local differences on farms, companies and so on. There'll be different breeds of birds. The free range are sort of longer living and so on. So I think the same still applies. These are still lineages that you would see in both types of production system, but you may see some nuances in the different sequence types profiles, and in particular antibiotic resistance. That's a really, really interesting question, and I would like to look at that. Yeah,
I think it would be. Well, thank you very much.
Welcome.
Thanks, Ed. Robin, I can see you've got your hand up.
Thanks, Fran. That was very, very interesting indeed. Two questions from me, and I apologise. I suspect I might have missed both of these, so sorry if I did. But the first one was, is there any consistent change when you talked about the introduction of new measures to reduce Campylobacter, and that's reduced, in particular, the density, right, of Campylobacter on chickens? I'm wondering if there's any sort of pattern in terms of the shift in isolates after that? So has that regime favoured certain isolates over others, or did you see a general reduction right across them? And then the second question was on that. I think you said that one isolate in particular that shows real seasonality as opposed to some of the others. Is there anything in the genome that might explain that? Is it more sensitive to temperature or desiccation or something?
That's so interesting. So for your first question related to the density of the chickens, I haven't seen an effect on the sequence types that are coming through. I think it's a general dampening down. Like I say, I'm doing this as a proxy through the human disease isolates. So I think that would be an interesting question to look at in the retail sort of abattoir-based surveys also. But that's the impression that I'm getting. It's a general effect. And secondly, the ST45 clonal complex that's more predominant in the summer, that's a really, really interesting point, and I really don't know. But you would presume there's something about that makes it both able to adjust to different hosts, environments and different gut physiologies and so on. Sometimes diet can play a part, and maybe it's more adaptable. So, for example, Sam did some work with a cattle-associated strain, and well, I need to double check this, but there was something to do with vitamin B12 or something like that in the host diet that affected the lineages. But so maybe ST45 is just more adaptable that way. But I'm just thinking on the spot here. I mean, it would be interesting to look at those genome-wide association type approaches and see if there is anything that we can predict and then test in the lab. But I think it's a really interesting question.
That's a really interesting point about diet. I hadn't thought about that. I mean, I guess one way to test that might be to look at whether ST45 shows a different seasonality in housed versus free range flocks, because presumably the free range flocks, they're changing their diet, but they're outdoors much more in the summer, whereas the housed flocks are getting more or less the same thing all year round. But just a suggestion. Fantastic.
Yeah,
thanks.
no, it's interesting. Yeah, thank you.
Thanks, Robin. Yulia, I'll come to you next, and then I think Angela, you put a question in the Q&A.
Hi, thank you for your talk, Frances. That's very interesting. We're doing some campy work at the FSA at the moment as well, so it's really good to see your presentation. The question I was going to ask is that you've observed as we have that the level of campy on chicken, so the contamination rate seems to have gone down quite massively in the last several years. But I've noticed that in terms of disease rates in humans, that seems to have remained stable. Do you have any hypotheses that you think would explain that trend, since we think a lot of the campy from humans does come from chickens?
Yes, good question. And this, I think, demonstrates the importance of having these longitudinal studies to really see whether the interventions that we're putting in place are helping. And I think from the end point, like you say, there hasn't been much impact, and I suggest it's a numbers game, probably, that you need to reduce it further than what it is. It's still chicken types that are coming through, and there's still enough, I would suggest, to be causing the amount of disease that there is. So, I mean, there's been some very positive progress there. But like I say, I think these types of studies where we can understand a bit more about how they survive the processing stages. For example, there is one clonal complex that I do see commonly in chickens on farms that I don't hardly ever pick up in the human disease. So I think there's some variability in the survival rates once they're outside the host bird, and then how they go on to cause human disease. So yeah, it's interesting.
We might be in touch with you a bit later if that's okay, because we're trying to unpick that question, and I think your experience would be very valuable in this. Oh,
brilliant! Please do. No, that would be really, really good. And I also meant to say with Campylobacter or any of the other organisms that we have on PubMLST, we're able to set up sort of projects. They can be private, restricted access if necessary, or public, as this one is, so that all of the information is on there, and analysis tools and so on. Sorry.
Great. Thank you, Yulia.
Okay.
Brown moment.
Thank you.
Thank you very much. Hope you're okay.
Sorry, my voice is giving out.
But I can get in touch with you, Yulia, after this and to pass on Fran's email address.
Yeah, that'd be great.
You can have a little chat.
Thank you.
Okay, Angela, I'll come to you next if that's okay.
Hi, Francis. That was a great presentation. Thank you. I just had a question. You might not be able to answer it. Just about the disease isolates that you got from the hospitals. I just wondered if they represented like the demographic of the UK as a whole. I don't know whether you know that, and just if you knew if there was any correlation between consumer behaviour and the Campylobacter. So we've been looking at things like people washing chicken and how they behave in the kitchen. I don't know if you are able to answer that at all.
Yes, that's a really interesting question. So what we're seeing in Oxfordshire does represent what we're seeing nationally, those sequence types that are coming through, and that's why we take all of the isolates that come through rather than sort of cherry pick, because we want that population overview. And we've tested that formally using different sentinel surveillance sites in the northeast of England, and we also did some work with hospitals in Hampshire. So we've tested all that, and basically what we're seeing in Oxfordshire is kind of 1/100th of what we're seeing on the national scale. Also in that previous published report, covering the isolates 2015 to 2018, there was a questionnaire that went alongside that. So you might be interested to have a look at that. And that was looking at things like foreign travel. I can't remember, I don't think washing chicken was in there, but it's certainly really, really interesting. I think there was things about where they bought chicken. I know sometimes there can be effects of eating out. If people don't eat chicken at home regularly and then eat it when they go out at the restaurant, sometimes they're more likely to have an infection. Perhaps they haven't built up immunity or so on. I've seen that as a slight effect. And there was a study by one of my colleagues, Noel McCarthy, who is, if people were doing sort of wild water swimming, which I know is sort of gaining popularity at the moment, they were more likely to get some of the environmental type Campylobacters. So you may potentially see an effect, and I think that's really, really interesting work.
Thank you for that, Francis.
Thank you.
Thanks, Angela. And Ed, I'll come back to you. You've got your hand up.
Thanks. Just another one that occurred to me, just based on something you said at the end about working with pet owners, I think, and you had a little slide earlier about kind of companion animal associated Campylobacter, and whether you looked at that and whether you had any information about what the owners were feeding their pets. I'm thinking specifically about raw pet food there.
Yeah, that's a really big concern, isn't it? This is work, the sort of planning is ongoing. So it's an area that I think we need to look at, particularly with the raw pet food. The sort of things I'm also interested in is those backyard chicken collections and how they compare with what we're seeing in industry. So you've got opposite ends of the extreme of how they're kept, what they're exposed to. So not being disrespectful, but on farms they have very strict adherence or policies for biosecurity, which wouldn't be applied to backyard chickens, sort of pet chickens. So I think that's an opportunity to see how Campylobacter behaves in different situations. And with the pet collections, we don't have the information back yet from those to be able to answer your questions. People have looked at this on and off, and often they are sort of those common clonal complexes that we'd be familiar with, and does appear to be coming through pet food to the pets, as far as I know. That's the most likely route for their infection. Also in dogs, there's Campylobacter upsaliensis. I think that is a bit more common, alongside Jejuni and coli.
Cool, thank you very much.
Great, thanks, Ed. I think that's all the questions that I can see. So I think, and given the time, I think that's probably a good place to wrap up. So just a couple of things to kind of close off the session. So in terms of where things are at kind of more broadly for the programme and kind of just now and into the future, projects like Workstream 2D are currently in progress and due to conclude in March this year, but we hope that it will be possible to secure further funding to continue the work, and there's a number of kind of avenues under exploration at the moment on that front. If people are interested in hearing more about the progress of PathSafe as we move forward, we share information regularly in a number of different ways through newsletters, via the website, etc. So if you, I would say to check out our website for more information, and of course reach out in the interim if you have any questions at all about the programme or more questions on Fran's work. So we're likely to take a break from the webinars in February because we're in the midst of organising a programme conference, but we aim to look to hold another one in March, so look out for more information on that. So finally, thank you to Fran for such an excellent talk today, and all the attendees for your great questions. So thank you all and have a wonderful rest of day.